I guess I'll start by saying that I was not in Port Alberni. I was just 10 minutes away from Port Alberni, where it was a little bit nicer, maybe. With a nice house with a pool and a hot tub and the river just by, we could snorkel with salmon and stuff like this. So it was really a nice introduction to Vancouver Island, I think. And Bamfield is just as beautiful, if not more beautiful, than Port Alberni, I guess. So I want to walk you through a list of different projects that I've been working on over the past few years, but generally around the theme of how ectothermic animals are coping with mostly extreme heat, but generally global warming. And before I start, I want to introduce you to this anecdote of the boiling frog, if you're not familiar with this one. And it says that if you put a frog in a pot of boiling water, the frog will immediately jump out of the pot. But if instead you slowly increase the temperature of the environment, the frog will not sense the danger and eventually be cooked to its death. Well, if you're a biologist, especially a thermal biologist, you see that this anecdote is completely wrong. And you shouldn't try this at home, even if you're French. And because if you try to put the frog in a pot of water, boiling water, the frog will not be able to jump out and basically die almost immediately. And if instead you increase the temperature of the environment, the frog will perceive those changes and eventually escape when the conditions get uncomfortable. But I kind of like this anecdote because it reminds me of the way we measure heat tolerance in the lab for a lot of ectothermic animals. So ectotherms are basically 99% of our animal biodiversity. They're like the fish, reptiles, amphibians, invertebrates, all of these really beautiful creatures that really make us quite excited about biology quite in general. But they're quite special in the context of climate change because their body temperatures are directly linked to environmental temperatures and all of their physiological processes. So it means that potentially climate change may be quite important for them and affect a lot of their physiological processes. And the way we typically measure heat tolerance in ectothermic animals is a bit like the boiling frog anecdote, where we ramp the temperatures up progressively until the animal, could be a frog, could be a fish, stop being able to maintain their physiological functions. So in a fish, it's usually a loss of equilibrium. In a frog, it will be an inability to be righting itself. So there's all of these behavioral responses that are usually termed CT max. You probably have seen this term, or critical thermal maximum. Just remember, this is the maximum temperature they can tolerate over a short period of time. And I've been very interested in CT max in general because it's been studied for decades. So we have hundreds and hundreds of publications measuring CT max across a wide range of species. And traditionally, it's been also used to try to link these heat tolerance limits to the temperatures organisms experience in a natural environment. So we can measure climate vulnerability basically as the distance between environmental temperatures and CT max. And that's something I've discovered during my master's, where I was reading publications from Jen during my master's degree, and we were just reviewing those and criticizing them a bit like you were doing with, I think, my paper also today. So, that was just really, really cool exercise, and it kind of got me interested in working on this topic. But what I'm mostly interested in is what's happening as the environment warms. How do environmental temperatures shift, and how CT max also shifts through phenotypic plasticity, and whether this phenotypic plasticity allows organisms to cope and buffer these changes in environmental temperatures. And I'm going to start with this first study that we were super lucky to publish in Nature. I don't know how it happened, but apparently it did. So it was kind of the culmination of my PhD, where it integrated various different projects and tons of people involved throughout the process. It was about 3.5 years of work. So it was quite a lot of different things and a lot of problems that happened along the way. But it was also the project that taught me the most, I think, as a biologist. I've learned so many skills and so many ideas. And it was really challenging, but very also rewarding in the same way. And the idea was to try to quantify at global scales how amphibians are coping with climate warming. And we wanted to have this really global and comprehensive understanding of their resilience to environmental change. And so the first step was to try to collect as much data as we possibly could find on the CT max, these heat tolerance limits of amphibians. We got about 3,000 estimates from 600 species, so it's really good data set. And what's pretty special about this paper is that none of the people on the author list knew what CT max was before they started these projects. So they're not thermal ecologists. My supervisors didn't know what CT max was either, and I kind of convinced them that it was kind of interesting. And the idea here was to just get non-native English speakers to try to retrieve data from the non-English literature. It turns out about like 12%, I think, from the top of my head, of studies were published in non-English language literature. So it's quite a huge amount of data that we tend to overlook usually in data synthesis. And it was really fun also to work with people outside of your field. I think I'll recommend this to anyone. It's really nice to see the perspectives and some of the different ideas they bring also to the project and how we can approach those problems. So now we have a bunch of CityMax data, and I was really happy about these data sets until I put it on the map. And as you can see, there's huge geographical biases. It's not new to any of the ecological data sets we have. They're usually as patchy as this one, if not more patchy. And we have entire areas of the world, usually areas of the global south, for which we have no or very little data. What gets worse is that, well, I guess I should mention also that the non-English literature points, I don't know if you can see them with these colors, didn't really improve that much the geographical coverage, although they allowed us to get some additional species we haven't had before. What gets worse is when you try to overlay these data sets with where amphibians live. And you see this huge mismatch between where we tend to collect the data and where amphibians are the most diverse and abundant. So that means that we know very little about where most of the diversity is concentrated and where potentially they are argued, usually in the literature, to be the most sensitive to environmental change. So that was quite disappointing. This is a representation of my face when I put this plot together. I was really disappointed. And I really thought that it wouldn't be possible to do this original project I wanted to do. Then I looked at the taxonomic coverage. Here we covered about 7% of described amphibian species. Doesn't sound like a lot, but it's still quite a lot of data, but probably not enough if we want a comprehensive assessment. And we have data that we can barely see, but pretty much all across the tree. But there's very big chunks missing in some families of amphibians and some particular clusters. And if you don't know what these guys are, they're called Sicilians, and they're these weird worm-looking like creatures. And it's an entire order of amphibians for which we have no data on CityMax. So out of the 3 orders of amphibians, we have 1 missing, which is pretty disappointing. And as far as I know, nobody knows their CityMax. So to summarize, we have both taxonomical and geographical biases in sampling. And that really calls into question whether we could assess that broad general question. And I see potentially 2 solutions. Either we could just analyze the data set as it is and just acknowledge its limitations. We know that we may not be able to predict what happens in those biodiverse regions, those areas that tend to be very undersampled. We could also acknowledge that in those particular areas, or to just get broad general patterns, some of those biases may bias also our data sets. Or we could try to solve these gaps. And that's what we've done. We developed this statistical method called data imputation or phylogenetic data imputation, where we're trying to retrieve and predict what will be the CityMax of those data deficient species. And so the way it works is that it uses the knowledge we have in our data set. So we have the CityMax, the heat tolerance of many of those species. We know that CityMax is phylogenetically clustered, so related families tend to have very similar CityMax. And we also know a bunch of other traits that we know are correlated with CityMax. And because it's been studied so heavily for maybe 100+ years now. And then for the data deficient species, although we don't know their CityMax, we still know a few things about them. We know where they live, and we know what temperatures they experience in their environment. We know where they sit in a phylogenetic tree, and we also know a bunch of those traits that we know are correlates of CityMax. So what we can do then is to train our model to predict what would be the CityMax for that given species, given all of this combination of traits. And we could do this for all the species we can find enough data for, which is about 5,203 species, which is about 60% of the amphibian biodiversity. And so we basically multiply by 10 the original taxonomic scope of our data sets. And that sounds almost a bit too good to be true, right? But it seems to kind of work. Here we've done a cross-validation approach. So we took our original data sets, we trimmed some entries, and we rerun our imputation model to try to predict these real known experimental values. And we've done this many, many times, and this is kind of what we find. So this very strong matching between experimental and imputed data. Of course, there's always a bit of noise. This is biology. This is also a model, so it's imperfect. But I think it seems to be unbiased and probably give us a good estimate of what could be the CityMax of those data deficient species. And we have this for a lot of species, so then we can try to look at how much temperature they're experiencing in their environments. And usually animals can really have different strategies to avoid the heat, and so their body temperatures can be very different from ambient environmental temperatures. So what we need to use is usually those biophysical models, which basically are solving the equations of life to try to predict what will be the body temperature of those organisms under different conditions. And so we simulated 3 different microenvironments that are quite relevant for amphibians. We have a terrestrial condition, an aquatic condition, and an arboreal condition. And in all of those, we assume 85% of shade. So what we try to do here is to simulate a frog or a salamander that is in an environment, a thermal retreat, basically, where they would be during an extreme heat event. They wouldn't be basking in the sun when the temperatures are too hot, especially if it approaches the maximum temperature they can cope with. And we ran all of these models for all species across the entire distribution range in 3 different climate warming scenarios. So basically had about 100,000 biophysical models. These take between like 5 minutes to like mostly 45 minutes to run. So you can imagine it was a huge computational effort that took many weeks. But we kind of managed to do it with supercomputers running constantly and a bunch of different fun computing and coding struggles, but it kind of worked in the end. So now we have the body temperatures of those animals in different conditions. We also have their CT max, and we could try to compare those values to see how sensitive they are to environmental change. Here is the temperature time series for that really cute little species of frog. And we also have their CT max here, which will be their maximum temperature they can tolerate. You can see that CT max is not a perfectly straight line. It's because we also accounted for plasticity. So every week, the animals are acclimated to the temperature they experienced before. So there's a bit of variation in CT max. But you can see it doesn't make a huge difference because amphibians tend to be very little plastic in general. And then we can measure how many times and to what extent body temperatures are predicted to exceed these physiological limits to understand how vulnerable they are to climate warming. And again, we've done this across different warming scenarios as well to try to understand the impacts of climate change on these organisms. What have we found? Here we have the predictions in terrestrial systems. I'm not going to present the arboreal one because it's quite similar overall, just to a slightly lower magnitude because we have less species. And what we found is this really huge increase in the number of species and the number of communities that are predicted to experience overheating events. This is the number of species that are predicted to overheat at least once. So it's not necessarily always a huge amount, but we can clearly see that there's very big areas of the world that are increasing, and there's some areas that are not vulnerable currently that become vulnerable, particularly in the temperate regions. What's also interesting here is that we see that the areas in which most species tend to be vulnerable are also the areas for which we have the least amount of data in the first place. So this data imputation approach is quite useful to try to identify where we should do some more research to try to validate those predictions, but it also calls into question how realistic those predictions are because we have so little knowledge about those particular species. So the goal now is to try to validate those predictions empirically and have people go in the field, collect the animals, and measure their CT max so we can improve that model at the end. Then it's really important to look also at how often these animals are experiencing those overheating events. So here we have the patterns with latitudes, and you see very clearly this escalation of overheating risk with climate warming. So this stark contrast between this 2 degrees and the 4 degrees warming scenario, between the yellow and the pink. And that really means that we need to reduce our greenhouse gas emissions as soon as possible. Clearly, once you pass this kind of tipping points, that's when a lot of species are starting to experiencing temperatures that are threatening their survival. And what's really interesting too is generally the patterns with latitude in species sensitivity are usually concentrated towards the tropics. So most tropical species are assumed to be more vulnerable to climate warming. But what we found is that it really depends. If you're in the northern hemisphere, usually subtropical species or temperate species tend to be more sensitive, whereas in the southern hemisphere, we have more tropical species being sensitive, which kind of goes against some of those previous patterns. And that's because we also accounted for those microclimatic temperatures, these biophysical models that allow us to go not to the air temperature, but to the realized temperature of those organisms. So that's pretty disappointing. Not disappointing, but a bit like sad to see that there's so much overheating events that are possibly going to happen in the future. The good thing is that very few species tend to overheat in ponds and aquatic systems. So it seems like these habitats are extremely important for amphibians to try to regulate their body temperature and survive the heat. And so it's really important that we protect and restore those habitats to allow them to survive in a warming world. But all of those predictions are probably a best case scenario. We know that we've assumed that they were in the shade at all times across the entire range of distribution and that it could maintain wet skin. But we know that those conditions are becoming less and less common. We have deforestation and droughts, for example, that are really impacting these habitats that amphibians need to thermoregulate. So it's very likely that all of those temperatures are going to be pretty big overestimates of what they would experience in the wild in some conditions. So take-home message from that study, the first thing is that these kind of statistical approaches are quite interesting. I think they can be used in combination to empirical efforts to try to solve some of the gaps that we have in data sets. It's really nice that it can allow us to generate predictions and actually get funding to perhaps do some research in those areas that tend to be less studied. And we could try to validate those predictions. But we found that amphibians will likely overheat in many, many areas of the world, and particularly in those biodiversity hotspots, which are the very areas that we tend to study the least. So we really need to spend a lot of effort towards trying to go and sampling organisms from those areas to really understand their sensitivity and their vulnerability to environmental change. We also found that the impacts of climate warming really escalate beyond 2 degrees of climate warming, and it's really important that we both reduce our greenhouse gas emissions, but also try to restore and protect those environments that they need to use to be able to regulate and survive in the changing world. Then second part of this talk, and I'm going to talk more about early life stages and embryos. This is something I've been very interested in since maybe the second chapter of my PhD, where I've done a project on developmental plasticity, found that embryos were quite sensitive, and now I've kind of directed my research towards comparing different life stages and trying to understand how they vary in their sensitivity to environmental change. But how much do we know about them? What we know is that usually environmental stresses have much higher impacts on early life stages. We know that embryos tend to be quite sensitive and other early life stages like young larvae, for example. And this seems to be quite common across the tree of life, from fish to reptiles to amphibians. They're usually a little bit more sensitive to environmental stresses. In the context of fish, they are a lot more sensitive, for example. And we also have some really nice hypotheses about what could drive variation in thermal tolerance or plasticity. I think it's a bit hard to see, the figures are a bit small. But this is a paper we published quite recently now about insects, because why not? But the insects have these very complex life history strategies. They go through metamorphosis, they are changing in morphology, in the habitats they experience. And this is likely to generate some variation in the temperatures they experience, and therefore the selection pressures on developing high thermal tolerance or high plasticity. So you have the examples, for example, mosquitoes or dragonflies going from aquatic to terrestrial environments, the cicadas going from underground to aerial condition, or some changes in diurnal activity, for example, that really fundamentally modify the environments that they experience in the field. So it's really important that we kind of look into embryos and the differences across life stages to understand how organisms cope with environmental change. So what we wanted to do here is to try to understand how much research is done on embryos, and often we hear that they are quite understudied, but it's more of a casual statement, and we didn't really have evidence for this. So we looked into 3 different journals, basically grabbed all the publications we could find, and we were focusing on experimental studies that were measuring responses to climatic stresses, particularly, and we're focusing on physiological and life history responses. And what I'd like you to do, if it works, is try to have a guess of what is the proportion of studies that are done on embryos relative to other life stages. And then we could try to see this together. And if it doesn't work, we'll just do a show of hands. Yes?
Is that proportion out of the 4,300 or out of the 1,200?
Just generally across studies, if you're looking at a study that looks at the impacts of temperature, oxygen, or other climate change stresses on physiology and life history, how many of the studies, the percentage of studies that look at embryos, embryonic traits versus other life stages? Yes?
It's waiting for your presentation to begin right now.
Interesting. What? It's not working then. I've had tons of issues with this thing.
You actually need to get the app.
Oh, okay, this is not gonna work. Usually it works. Maybe I don't have. I'm not connected to the internet or something, and it's not working. Let's do a show of hands. If you think this is less than 5%, okay, between 5 and 10%, between 10 and 20%. Okay, I may have biased also your opinion because I just said that they were quite understudied, but you're probably right. Oh, well, this doesn't work. This is meant to look like it. This is meant to. Oh, okay, 5 to 10%. So it somehow worked for some people. It's about 8 to 9% of studies that we found. And what's pretty interesting is we found that it's very consistent across all the journals we looked at, and also all the different categories we could possibly look at. So between taxa, between climate change stresses, between, I don't know, we tested a bunch of stuff. Between the type of traits that we're looking at, it seems to be always around between 5 and 15% at best. So they're generally quite understudied, and this pattern is very, very consistent. What's funny here is you might see that in the journal Conservation Physiology, for example, we had more studies in which the life stage was unclear than we had studies on embryos. So that really shows how understudied they are, I think, and that's a really cool example. I gave this talk at Society for Experimental Biology, where there's the chief editor of Conservation Physiology, and he didn't like that I made that comment, but I did anyway. Also, I told you that there's this growing recognition that embryos are, I'm still on amphibians apparently, embryos are quite vulnerable to environmental stresses, and I think this has been pretty well accepted in the community. So we expected to see an increase and an uptick in the number of studies that are done on embryos, but we did not find this at all. It seems to be very consistent, at least in the past 10 years, where there hasn't been really an uptake in the number of studies on embryos, although there's so much evidence and really accumulating evidence that they're quite sensitive to environmental change. So that's quite concerning. And I guess the take-home message from this is that we really need to study embryos a bit more. They're quite hard to study, though. Like, usually you can't find the eggs in the wild. We can't breed the animals in the lab. There's all of those challenges. Some of the equipment that we use cannot really work because they're so small and we can't really measure the trace that we want. But that's really what gets us excited, I think, as biologists, is, this thing that hasn't been studied at all. There's tons of opportunities, and we just need to crack that problem. So I think there's going to be a lot of development, hopefully, in the field, trying to understand the biology and the reproductive biology of these animals, try to make sure that we can keep them in the lab and breed them, and hopefully a lot of opportunities for research coming from there. At least I'm going to do this, but we'll see about the rest of the people. And that brings me to the research I've been doing at the University of Gothenburg, which has been really focusing on those comparisons between life stages, and more from the experimental space now. So I've done a lot of comparative studies and stuff, and now I kind of go back to the lab and also do some experiments and combine this with these comparative studies to try to get data that we don't have in the literature. And the idea is to really get a global understanding, a bit like the Amphibians project, but with fish and across life stages. I'm interested in fish because they are the most diverse group of vertebrates. It's over 50% of the vertebrate species. They have this unique diversity of shapes and forms, and some weird things with the background here, but okay. They're really cool. Like, they live in some of the most extreme environments on Earth. They've really colonized all sorts of habitats. And I think that gives us a lot of opportunities to study them and understand how they colonize those habitats. But it's also because of these unique specializations, they might be quite sensitive to changes in their environment. And that's what we find. If you look at the news every year, following a heat wave, in all countries you could think about, there are always some massive die-offs happening for fish. I was in Vietnam when this one hit, and I saw thousands and thousands of dead fish in Hanoi, in the lake that was nearby my hotel. And I can tell you it changes you as a human being to see these kind of impacts. And I think I have this kind of moral responsibility to try to better understand what causes those impacts to happen and how we could possibly develop some early warning risk or some tools that allow us to try to better understand why do these happen and how we can mitigate those impacts. We were near Port Alberni not so long ago, and the temperatures were quite warm. We saw a lot of dead salmon in the rivers as well. I don't know if it was connected to the temperatures, perhaps, but this may happen also in your backyard. So I think it's something that is very topical, and the consequences for our society will be huge if we keep having those that are becoming more intense and more frequent all over the world. What's even worse is that it seems like early life stages are much more sensitive than the impacts that we visualize with the adults and these massive die-offs. So according to that study in Science, embryos are a lot more sensitive to extreme heat. And if we just look at their embryonic heat tolerance, their CT max, 60% of fish species may exceed their heat tolerance by the end of the century. 60% means a complete collapse of all the ecosystems that we know of. It makes a collapse of our culture and society and a lot of the impacts that we can have. So this seems to be almost like absolutely dramatic for everything that we know. So I really wanted to see if we can study this a bit more, because this will have huge impacts on the world as we know it. I know I was talking to some of you about writing commentaries and things like this. So this is what I've done early in my PhD. I don't know if it's a good idea or not. I wouldn't recommend. But what I found in this study is that the methods that we tend to use to compare different life stages and draw comparisons specifically based on their heat tolerance tend to be very inconsistent in terms of methodology. So in adults and juveniles, we tend to use CT max, so this this increase in temperature progressively until the animals lose equilibrium. And so this is typically a trial that lasts about 45 minutes, maybe an hour in some species, and we measure sublethal response, so when the fish can no longer swim in response to the heat. Whereas in embryos, we tend to usually expose them from fertilization to hatching or even to first feeding. In salmonids, for example, this is months, and we expose them to those constant temperature for a very long period of time, and we measure how many of them die or do not hatch. So we're looking at a lethal response that is very chronic in that case. So you can see these things are almost like antonyms of each other. Like it's completely different methods that we use to compare those life stages. And because these ones are usually a lot more extreme, we expect naturally to see lower thermal tolerance in embryos compared to later life stages, sorry. So that really calls into question whether what we see and those huge differences in thermal tolerance between life stages are just methodological artifacts or really true biological differences in thermal tolerance. And instead of just writing a commentary and being a bit critical, I just wanted to test it myself and do some experiments. And I think we can draw from the research that we know on invertebrates to try to apply this to fish. So there's these new frameworks to try to measure thermal tolerance that are not CT max, and they're usually called in various different ways. I think it was originally called the thermal tolerance landscape. Now a lot of people like to call it TDT for thermal death time. Now there's a new paper that came out last year that call it thermal load sensitivity, which I like better personally. But this basically means that we're integrating not just the temperature, but also the duration at which this temperature is applied. And according to this framework, we can predict survival under any temperature time combinations. And that's really useful because we can integrate this with natural thermal fluctuations, so these temperature time series, and predict survival in these fluctuating thermal regimes. And natural environments are always fluctuating, so that's going to be very useful. And the general framework for this, to understand it, is that think about a sauna. The temperatures are usually between 70 and 80 degrees. We could survive there for like 10 minutes, maybe 20 minutes, maybe 40 minutes if you're Finnish. But you wouldn't be able to stay there for 6 hours. Like we will all die after a very long period of exposure. And the idea is this, to just measure how long each organism can tolerate different temperatures. And we could do this across different life stages. We could use exactly the same type of method and the same duration of exposure and try to compare those life stages using the exact same experimental method. The first thing we did was to try to replicate what people do usually in embryos. So we expose eggs from fertilization to hatching. This was all done in zebrafish. Zebrafish are actually quite cool in climate change biology because they're exposed to very, very extreme temperatures in their natural environment. So it's not just a medical system that is easy to take care of. It's also quite a nice system in experimental biology. And you can measure hatching rates, so the survival rates with temperature. And we can measure the median lethal temperature, so the temperature that will kill 50% of the individuals. And in zebrafish, it's about 36.2 degrees. That's much lower than the CT max that we tend to measure. CT max is usually between 40 and 42 degrees. So that kind of confirms this science paper showing that there's very large differences in thermal tolerance between different life stages. But again, it's really important that we remember the duration of exposure here. In zebrafish, this temperature corresponds to a development time of about 60 hours. So it's at least 60 times longer than the traditional test we do for adults and larvae as well. So perhaps that explains a lot of those differences. Then what we did is measure CT max. What we've done is to measure the behavior of embryos in the egg, and we measure the temperature at which they stop moving, and we measure the loss of equilibrium in larvae, which is a traditional endpoint we use. And it's been proposed to be pretty consistent across different life stages. And what we found is, again, something very similar to that science paper. Embryos are a lot less tolerant than larval stages, but the pattern we see with the rate of heating, so how fast we are ramping them up during the trial, seems to vary quite a lot between the different life stages. What it means that if you use a very slow ramping trial, that means the differences between life stages is huge. As if you do a very fast trial, we don't see very large differences in thermal tolerance. And then we looked at, okay, maybe those endpoints that we're using, the behavior of embryos versus the loss of equilibrium, maybe these are actually not really always the same thing. And we've done this thermal death time thing that I mentioned before. So this plot is a little bit complicated, but what you see is this, the time it takes for 50% of the individuals to die across different temperatures. And it just follows this typical relationship that we expect with this thermal death time, which is an exponential decline in survival time with hot temperatures. So you could thaw at 43 degrees maybe for a minute, but you could thaw at 38 degrees for 6 hours, for example. So typical what we would expect. And we used 3 different life stages this time because we could use younger embryos in which we can't really measure behavior because they're just dividing cells. We use also late embryos, the same as before, where they already exhibit movement, they have a little bit of pigmentation in the eye and so on. And the larval stage, again, same as before. Usually those plots are a bit easier to visualize on the log scale because it gets linear, it's quite nice and easy to compare. And what it means is the more the curves are shifted to the left, the less tolerant the animal are. So they can't tolerate, they will be able to tolerate this temperature for less long than at a higher temperature. And what we see is a 1.5 degree difference between these two clusters of life stages. But as you can see, it's completely opposite to what we found before. The late embryonic stage was less tolerant than the larvae in the previous plot, and now it's a completely reversed relationship. And what it means basically is that the endpoint we're using, this behavioral response, is not the same proxy of survival limits as the other cases. So we really need to look more into what response can be compared when we're using sublethal endpoints across different life stages. And this goes completely against that science paper because we found that embryos are actually a lot more tolerant than other life stages. But regardless of whether they're more tolerant or not, this shows that the variation that we see across life stages in this particular species are not huge. 1.5 degrees is much less than the 10 degrees that we usually see once we stop standardizing the methods. So that's kind of cool. There's a lot more things that I've done there, but it's going to get too complicated and boring for you, so I'm not going to show you this. Just take a message from that study is that experiments are pretty fun. We could do some cool stuff with across life stages. But also, once you integrate this, both the duration and intensity of heat, you can find very different patterns in which life stage is more sensitive than others. And I think that previous differences among life stages are likely to be entirely driven by methodological artifacts because we tend to expose embryos to much longer periods of time than the other life stages. So we definitely need a lot more studies using this kind of framework to try to validate and see if there are some cases in which embryos are more sensitive, and if they are not, why? And how can they tolerate such high temperatures? Because they don't have the same kind of physiological processes that can enable them to cope with the heat. They're limited with the egg and the diffusion of oxygen within the egg. So there's a lot of limitations, and they still manage to be extremely tolerant. Now I'm going to go into the last part of the talk, and I'm going to talk about something I've initiated in October called the Thermal Ecology Alliance, which is basically a call to try to make science more collaborative and try to, as you can see, like all the projects I've done, I haven't really highlighted all the people, but it's been always super collaborative, and I think that's one of the funniest parts of science is to work with people and learn from them. And this is just trying to take this to the next level and make collaborations a lot more inclusive and a lot more global. But there's some reasons why I've naturally converged towards trying to go with this. It's also because as a comparative biologist, we face a lot of challenge with the data we have available. And when we try to extract data, oftentimes it gets a bit difficult. And you'll see this in your ecological data synthesis course, that there's some variation among the different methods that people use. There's data that is patchy, data that you cannot access. So it gets quite complicated. So this was the first, my first PhD chapter. I looked at differences between sexes and how they can cope with extreme heat, particularly how they can acclimate to changing environments. And out of all the studies I found that were having the type of experimental design I needed for that particular project, 77% of studies were not reporting the sex of the animals. So I couldn't really extract that data for my project. So I was left with a very weak sample of studies that were super patchy and heterogeneous, and had to just kind of deal with what we could get, which was a bit frustrating. So it shows that we really need to make a huge effort in making sure that the data that we provide to the community is first accessible, but also I can't say this word, but you know what I mean. I think interoperable. Anyway, we need to be able to action these data sets, basically. Then the second PhD chapter, I looked at variation between life stages and how they can acclimate to changing environments. Doesn't really matter too much, but the data I was the most interested in was very, very poorly done. And then I spent most of my time basically accounting for the variation we have among those different studies. Everybody does a different project. They use different methods that they like. They make decisions that are not standardized or there's no consensus about. And you end up with a very, very messy data set full of variables that you need to account for. And you have a lot of time with the stats. So basically I spent 76% of the models that fitted to analyze this data were just about differences in methodology. And I wish I could spend 76% of my time doing biological questions because they're a lot more exciting. And that's what I call the curse of comparative biology, where we spend more time dealing with data sets that are messy than actually spending time answering biological questions, which is the thing that we really want to do. I mentioned this imputation approach and how it was really promising and really nice, but again, like, can we really trust those estimates without actually going to the field or going to the lab and validating those estimates? We always need to go back to collecting data regardless of what fancy stats we try to do. We also have these biases that I showed before, very pervasive, very, very common across all ecological data sets. We have these biases in sampling where some life stages, some species, some sexes are very, very understudied relative to others. So we're really dealing with a pervasive methodological heterogeneity and knowledge gaps that we have to deal with when trying to standardize and synthesize data from the literature. And whether you do this from the literature, or you're trying to just write a paper and see what others have done, a lot of the times you see those gaps and they really affect how we know the world and how we can interpret biology in general. And I think the Formal Ecology Alliance is kind of one step that can try to contribute to reducing some of these gaps potentially. And the idea is to build a global community of practice and a collaborative network where we try to identify these gaps and try to solve them together. We try to do distributed experiments and things at much wider scales, and then try to coordinate projects with consensus from the community before we actually action those projects. So far, we are 1,800 members, which is crazy. It's absolutely insane, which I emailed a ton of people, and that really works in a way. And we also got a lot of word of mouth. And I think it's also just recognizing this demand, I think, from the community to establish such network. If so many people are signing up from so many countries, and just the beauty of the emails I received about how excited people were to have a community where they could belong and do things more collaboratively, especially people in the global south that don't have these kind of opportunities. Oftentimes, they're not able to attend conferences. They don't really have the same opportunities to network with people. And that gives them a chance to actually collaborate and communicate with others, which I think is absolutely beautiful. One of the goals of Formal Ecology Alliance, is to foster projects, especially collaborative projects. But where it differs, I think, from other initiatives is that we allow anyone to participate. So you have the project that is open to the world and everybody can sign up. And then whether they become co-authors or participate to the project is depending on their contribution. So we can really allow these people in the global south to be able to participate in those projects particularly. And we also just make the project very open, so there's no duplication of effort. Everybody knows that this is happening. And I think this is a nice way because we always have duplication of efforts in some ways, and there's overlapping decisions. And we could also design the project as a community. I think all of these projects usually have little meetups and working groups to try to revise the experimental design, revise the question, and make sure that we all agree. And there's a round of peer review basically before the project happens, which is really nice. What's really cool is that anyone can propose projects. So if you have ideas and you want to just put this into the network, I'm happy to facilitate this. And this is not my network. This is for the community. So I'm not involved in all of the projects. I'm just here to just help people collaborate. And so if you have ideas and you just want to foster through this, please just, there's a form, you just put it on and I'll just work it out and I'll put a page on the website, basically. And so some of the stuff we're doing are these distributed experiments. So again, it's trying to tackle those methodological heterogeneity that I talked about. So we try to use standardized protocols that are consensus with the rest of the group and replicate the experiments at global scales or semi-global scale. And I think that's a really unique way to do research, because first we get the community engaged, we get the scientific knowledge, and we work together to design a project, and then we apply it, and we can know exactly where the gaps are and try to maybe reach out to people that could solve some of these gaps. So it's a bit more proactive than trying to get the data from the literature that is usually patchy and hard to access. So for example, we have one on fish embryos. So I was talking about this, and it's basically replicating the experiment I've done on zebrafish across other species and across the globe. So far, we have 95 people, which is phenomenal. I'm super excited. 37 countries and tons of different species. Some people are doing multiple species as well. So we really could get a very big data set. And it's super, super exciting. And if you want to do a distributed experiment, my advice is to make it as simple as you possibly can. If you do a really large experiment that will take 6 months, no one is going to do it. And the success of distributed experiments, they've been done in plant ecology quite a lot. People have done a lot of field work and done all of these things. But in experimental biology and animal ecology, this is really not common. So trying to initiate this and hopefully make this happen. But the feedback I got from the plant ecologists was just make it as easy as you can, like a 5-day thing, 10 days maybe, and then that's it. Otherwise, it's not going to work. So the idea here is to just do a very simple experiment that can be done in about 3 days per replicate. And then it's very simple, it requires little equipment, it's not too expensive. And we just rely on people spending time. And that's already a huge value, I think. And we're super grateful for people spending their time doing this. And then there's wide co-authorship with a lot of people. And we've had people doing this in Brazil, this is on the Pacific Islands, this is in the US. So it's been a good uptake. And you see that the setups are a bit different too. It's the same experiment. So we can't really remove all this methodological heterogeneity. It's just not realistic. All the species have different ecologies. They need specific setups to be able to be kept in the lab. So there's always some variation, but we try to reduce the variation as much as possible. So we just have this very standard design, and then people just try to adapt it to make it as comparable as possible to the rest of the standardized design. We also have a bunch of events. So there's some brewing ideas. So this is the kind of stuff where we're going to try to have these working group discussions. We have these Spill the Tea seminar series. So if you want to give a seminar, I'm happy to do this as well and organize this. And then some in-person workshops, hopefully, and some more educational seminar series. But ultimately, it's just really about building a community. It's about trying to get people together to think about how we can work together. And we're dealing with very, very big problems. Like climate change is accelerating, and we really need to get to those questions and try to solve this problem. And I think this is only possible as a team. So I'll put the QR code at the end if you want to have a look. But I just want to go back to this metaphor of the boiling frog. And I told you there was kind of completely irrelevant to the biology of animals. But I think that to define us, it's a pretty good metaphor. We are experiencing very dramatic changes in our climate, and I don't think our policies are coping with the magnitude of those changes, and we're not really acting at a pace that is matching this environmental change. So to me, we're just like the frog in the pot of water, slowly reaching a boiling point and being completely oblivious or ignoring a lot of those impacts. So I think it's time that we jump out of the pot. I think it's time that we just do everything that we can to try to tackle and predict and mitigate these environmental changes so we can protect the biodiversity that we care about. And I think these kind of global collaborative efforts are one way that we could try to team up and unite forces to try to tackle these big challenges. And I think this is the only way to tackle these big challenges. I've been working with a lot of people, and it's been the best. I couldn't put them all actually. So there's been a lot of people involved throughout the process, and I've been mentored by some of the most generous and exciting scientists I think in the world. It's been just a joy to learn from so many people across different fields, across different various levels of expertise, and I've been extremely grateful for all of this. I've been very lucky also to work on some beautiful places. I was on the land of the Beagle people in Sydney, for example. Now we're on Badfield, so it's also a traditional land. I think that needs to be acknowledged. I'd like to also acknowledge my privileges. I'm a white guy. I was born in France. Had access to free education and a bunch of other privileges that are not accessible to many in the world. Also, I just got a grant, so I'm going to recruit a PhD student and a postdoc. So if you're interested in working on these kind of questions, it's very broad, I think, in scope. So please get in touch, and I'm happy to talk about it with you as well. And yeah, I think I'm out of time anyway. So thank you for your attention. Hope you enjoyed it, and I'm happy to take questions.